This is going to be a answer key video for quiz 4. So we'll just go through this, and thanks for watching this. I make these to help student learning, and we've talked about this several times. So where in the target cell is the estrogen receptor located? The answer is the nucleus. The estrogen receptor is an unusual receptor. Most receptors are in the cell membrane and bind to the signaling factor, whether it's a hormone or paracrine factor, in the cell membrane. The estrogen receptor is not. The estrogen receptor is in the nucleus because it serves and functions as a transcription factor. What's the general term for a cell that responds to a hormone? We call those target cells. And there are lots of target cells for the estrogen hormone in human bodies and mammalian bodies and vertebrate bodies. The estrogen receptor is a member of what gene family? Only a couple of students got this. It's the nuclear receptors. And so it's important to appreciate that in us, when we talk about genes, very often they're members of gene families. And so it helps some for us to organize our learning in humans and mammals in general. Well, humans, let's stick with that. There are 48 nuclear receptors, and they're really famous: estrogen receptor, testosterone receptor, and so forth. And so it's a member of a gene family made up of paralogs that we learned about. So genes, two genes of the same species that are understood to have shared a common ancestral gene. So it's like this, set of tools that get to be kind of replicated and very similar, but actually playing distinct functions. Testosterone receptor, estrogen receptor playing different functions in our bodies. What's a paracrine factor? So it's a local signaling factor. And everybody did a great job on that. What are the source cells that make estrogen? And they're in the ovaries. That's a great answer. They're actually called the granulosa cells primarily, but ovaries is a great answer. Okay, so then just two drawings. And so the first drawing looks like this. So we're going to just have a source cell, and we got a label. So that's a source cell, and then a target cell. And the signaling factor is going between them, floating between them. And then this is where it gets a little bit challenging, because when we started learning about receptors, we learned about the estrogen receptor that's inside the cell and inside the nucleus, because it's a transcription factor, and that's where transcription factors do their job. Importantly, there are just those 48 nuclear receptors in human, and there are many other receptors, and they're almost all in the cell membrane. So we want to put our receptors here. Yes. We spent some time learning about estrogen and estrogen receptor just to get to the bigger picture of receptors. And so these are not nuclear receptors, and we'll learn about other receptors, and they're in the nuclear, and they're in the cell membrane. And how do we know this one is in this cell membrane? Because we need to write signal transduction pathway. So that's a series of biochemical steps here in the cytoplasm, and so that's where that label goes. And so in a sense, it wasn't a trick question, but it was a challenging question because of that, to recognize, well, if we're going to have a signal transduction pathway, we need one because the receptor is in the cell membrane. We got to bring the message into the cells. And you may have heard of the word, and this is something we haven't talked about, but you may have heard the word in other courses, especially intro bio, of second messengers. And second messengers are smaller molecules inside cells that play roles in signal transduction pathways. And to bring that up, just because in a second messenger, the label is because the primary, the first and primary signaling message is here. It's the signaling factor, and it stays outside the cell, and then these second messengers are parts of the signal transduction pathway inside the cell. So just an important clarification, sometimes in these kind of formative assessments, an instructor will write a question where it's just a little bit challenging. and we want to make it very straightforward, but it's important to, kind of challenge the students and help them appreciate that when we're talking about something that has a signal transduction pathway, the receptor must be in the cell membrane. Another way to say that is when the estrogen receptor is involved, there is no signal transduction pathway because the signaling factor estrogen is allowed to come right into the cell and bind directly to the estrogen receptor. Okay, so if you have any questions about that, let me know. And then here. Now, the first thing is I gave an example, and it wasn't a very complicated example. We talked about estrogen target genes, and here's one, and there are many, and one of them is vitellogenin. And vitellogenin is found in other vertebrates, but not in mammals, because the vitellogenin protein is called a yolk protein. And the female in every vertebrate except mammals, and except the mammals that are not marsupials, because we have some egg-laying mammals, so like a duck-billed platypus and echidna are mammals. And I just say, not in mammals, but there is that exception because they lay eggs. And so they have this. And yolk proteins are sources of nutrients. So what happens is mom makes these yolk proteins, and they just chain together a whole bunch of amino acids, and then in the embryo just breaks these down to the individual amino acids and uses the amino acids as a source to build the proteins it needs. So yolk protein, vitellogenin, is an estrogen target gene. So the gene is present the whole life in the female non-mammalian vertebrate, but it only gets activated when the female grows to become reproductive age, and because it's going to have to start making eggs, right? Like that, okay? And so it's going to activate the gene then. So it would be inactive in the juvenile and active in the adult. So inactive in the chicken baby, the young chick, and active in the adult hen, okay? And we're just adding that as an example to put it in a little context here, even, in the context of a quiz, because it's just a target gene, and we're just putting a name on the target gene. And so here's what we got to do. So the first one is to draw it in the chick, baby chick. And I'm just going to draw the nucleus there, so we should label that nucleus and to make the larger cell, but there it is. And we've got this. And one thing is I draw these genes like this, and I don't always label them, but really you should, to show that you understand it, because a couple of students labeled it incorrectly. So that's the transcription unit. This is called the promoter, and this is called the enhancer. And you should be in the habit of labeling those and be readily labeling on a test, because you just have to lock in on that. It's not going to be, changing, over your lifetime or anything like that. And we just have to appreciate that this is the regulatory region here, and that's the transcription unit there. And the regulatory region is made up of these small, discrete regulatory regions. And so this is where the estrogen receptor is going to be bound. So there's the estrogen receptor, and we've talked about this in a baby chick, in a juvenile animal, when there is no estrogen. So you don't want to draw estrogen in the baby chick. The estrogen receptor is there, and it's actually repressing gene expression. So I've talked about that a few times in class, that it's repressing, and in the video, it's repressing gene expression. So it's important to have it there. It's supposed to have it bound to the enhancer to show the organization of a gene. And we just had to be able to draw genes like that every time. Transcription unit, promoter, enhancer. And we're talking about these big transcription factors that regulate, big because they're important, not because they're especially large, but important transcription factors that are really the switches. Okay, so then what's the difference? The difference is that when we draw the exact same thing, and again, I don't bother to write it in, but you should, transcription unit, to show, and every time you call something, you strengthen your learning of it. So you should want to write these labels down to recall it and further your learning. So it's still going to have the estrogen receptor, okay? So, and want to label that and so forth like that. And again, a lot of times I skip over the labels just because they're implied, but the student, I should do it every time, right? To show the students. Here, in the hand, we want to show estrogen and label that and show that it can come right into the cell, and this is again the nucleus, and right into the nucleus and bind to the estrogen receptor. And what it does, it now activates the estrogen receptor. So it's no longer a repressor, it's now activator. And we want to draw this in. We're going to get RNA made, and it's going to be mRNA, so lots of molecules of mRNA. And then out here, that mRNA outside of the nucleus is going to bind to a ribosome. And we're going to have lots of vitellogenin protein present. So you can detect that in experiments. Yes, the DNA is detectable in the young chicken, but there's no detectable vitellogenin mRNA and no detectable vitellogenin protein. So those are experimental results to confirm and provide evidence for this model. Great. Okay, and so with that, we're all set. But I want to add one thing, and that is, right, so all vertebrates have the vitellogenin gene. It plays an essential role in making yolk proteins, all vertebrates, but then mammals don't, because mammals don't make a big egg. Mom doesn't provide nutrients in mammals by making a big egg, with the exception of marsupials like the duck-billed platypus and the echidna. They lay an egg, and mom puts vitellogenin in that. Why don't mammals make vitellogenin? Because the egg is small. Why is the egg small? Because it's going to implant in the uterus and get nutrients directly from mom through the uterine wall and eventually the placenta. So to help you remember that, here's just a piece of information that these genes were lost in mammals when the mammals started being able to nurse their babies, and also when they had a placenta. And one way to think about this vitellogenin gene in non-mammalian vertebrates is like this. So there are some non-mammalian vertebrates that make a big egg fast. So lots of species make eggs, and in some cases they make it fast. And so, for example, reptiles don't make it particularly fast because they are going to lay a series of eggs, so there would be probably some reptiles that make it faster. But there are a lot of fish that make pretty big eggs really fast. And so when that happened, you can see that the vitellogenin gene inadvertently went through gene duplication. And these, we'll say certain fish that want to make a lot of yolk protein fast, they actually duplicated their gene so they could make lots of it fast, right? This can make yolk protein, vitellogenin, 3 times as fast as an ancestor that just had one gene. And so it's a little set of paralogs, a little gene family, and the genes haven't really diverged very much. They're all doing the same thing, which is just making nutrients like that, proteins that'll be nutrients. Okay, so that's just one note. Then here's the next note, is that mammals, when they first evolved, had these genes, but they quit using them. And when they quit using them, a random mutation would occur. They didn't need them, so you get a random mutation. You can still find the gene back if, a couple hundred years ago. If there were a scientist back then, they could have found the gene, but had a mutation. And then another mutation and another, just random mutations. The gene's no longer being used, so it's going to disappear. But a key concept is that it disappears slowly. It's broken, it's a null allele, and it just builds up more and more mutations, but slowly disappears. And there's a term for that. And the vitellogenin genes in mammals became pseudogenes. So there's a new term that we're only going to mention probably once in our course, but it's an important concept in biology, and that is sometimes you can detect pseudogenes. Now, it was a couple of 100 million years ago when the mammals started to evolve and started to lose their vitellogenin genes. And so those pseudogenes are really almost no longer detectable. And here's just a little image of this. So common ancestor, and the chicken maintained vitellogenin gene, and then humans and other mammals, but just to make a direct comparison, started losing it. And we can still see remnants for that. Now, it turns out that the remnants were just really minor pieces in humans, and more recently, scientists decided they're probably not realistically. They're just so broken down that it's not really as realistic to call it a pseudogene in humans. It's really kind of gone because it was so long ago. On the other hand, there are examples of some other mammals. So, for example, marsupials had multiple vitellogenin genes, and they just have one they still use. And that's because they make the egg slowly, I'd say. And they've lost these others, and they've lost those more recently, and you can still find those. So pseudogenes, and it's just an important concept. And why is it an important concept for us? Because notice that when we say, what's the definition of a gene? We say a gene is physical, and it's composed of DNA. And it is made of two parts. And those are just so important that, we want to kind of emphasize those for in our course, because one part is the transcription unit, which is the functional part, the business part, the part that's going to be output and be the product. And then the regulatory region. And anytime we say regulatory region, we should be thinking about the promoter and enhancer, and we should be thinking about the fact that regulatory regions regulate where, when, and how much of a gene gets made. And then that's a really great definition for our course, but we also want to say that a gene affects the phenotype. And that's because pseudogenes do not affect the phenotype. They're not real genes. They're really broken down genes that can still be found in various species for various reasons. That their ancestor long ago had it, and they're in the process of just slowly losing it. Just because they've lost its function long ago. It's useless, but just to have it completely disappear from the genome takes a very long time. Okay, and that's it. Okay, so just a new term, a term just to kind of follow up on the use of that example of vitellogenin. Okay, great. All right, that's it. Thanks. Let me know if you have questions.